Okay, here we're entering our unit that I'm calling Pro Cell Structure and Function because it's kind of an introduction to the different cell types, and we're going to concentrate primarily on the simplest cells, and those would be prokaryotic cells, and the structures that all cells share. So just a review here, because we've looked at this before. Life began with the first cell. In other words, cells, the smallest thing that's considered to be alive is a cell, and we didn't have life on this planet until this first cells evolved. So the cell is the basic unit of life. All living things are composed of cells. Cells only come from other cells. So all the cells making up every single living organism on the planet today came from cells that lived in the past. And in multicellular organisms, if you'll recall, because again, this is review, cells make up tissues and organ systems and the whole organism. Your body's composed of trillions of cells, and there are about 200 different cell types that are specialized to carry out different functions. So understanding cell structure and function is fundamental to biology. You can't study biology without studying cells and understanding cells. And the discovery of the cell depended on the invention of the microscope. We didn't know that all living things were composed of cells until we had a microscope to be able to zoom in on the microscopic world, because cells are so small, and see and look at tissues from every single living thing and realize that everything is composed of cells. So what we're looking at here is a leukocyte, which is a white blood cell, WBC, white blood cell. And this is a colorized transmission electron micrograph, TEM, transmission electron micrograph. And it's colorized because electron micrographs are grayscale. Electrons cannot confer color. Color comes from light, different wavelengths of light that bounce off of whatever you're looking at to give them the color that they are. And electrons don't have that property, or they don't have that ability to confer color. So electron micrographs are always grayscale. And if there are colors, they have been assigned, and usually computer assigned, different colors based on the shades of gray. And so what we're going to do with this cell is identify the different parts of the cell that are common to all cells. But also notice that this cell is specialized. It's a white blood cell, and it's specialized to engulf invading organisms. So this cell could actually eat a bacterial cell. And if you look around the outside edge here, you'll see these little extensions that are known as pseudopodia. And it would use these pseudopodia to engulf and take in and eat, basically, bacterial cells and other foreign invading cells. So, all cells have a plasma membrane. No matter how simple the cell, it has a plasma membrane, the cell membrane that surrounds the entire cell. And again, in this cell, that's these extensions that known as pseudopodia are extensions of this plasma membrane. All cells have a cytoplasm. The cytoplasm is basically everything within the cell but outside the nucleus. The liquid portion is referred to as the cytosol. So you need to be aware of that term because you'll see it used, and you need to know that it's basically just the liquid portion of the cytoplasm. There are also organelles that are suspended within the cytosol. So the cytoplasm refers to the cytosol and all the organelles that are found within the cytosol. And finally, we have the nucleus. But not all cells have a nucleus, and that's one of the things that we'll be identifying in this unit. Cells that have a nucleus versus cells that don't have a nucleus. Cells that don't have a nucleus are the considered simpler cells, and they are the prokaryotic cells. They're prokaryotes. Cells that do have a nucleus and membrane-bound organelles, as we say, are eukaryotic cells, or eukaryotes. So we're looking at here the discovery of the cell and the development of this concept known as cell theory. And it involves 5 scientists. So you need to be aware of these scientists and what they did. And our first scientist is a guy named Robert Hooke. There he is right there. And he was looking at tree bark, basically, under the microscope, cork. And in looking at the cork, he noticed that it was divided up into these little units. And he didn't know what they were, and he decided to call them cells. So he was the first person to coin the term cell, and that's why we call cells cells today. Because to him, they looked like the little chambers that the monks in a monastery lived in, and those were referred to as cells. So he simply called them cells. So this is the kind of microscope he was using. And it's a relatively primitive microscope, but it allowed him at least to see the cork cells. And by the way, cork cells, plant cells in general are relatively large. So it was kind of lucky that he was looking at cork and he had relatively large cells to see, because this kind of microscope doesn't magnify that great. But he's basically the father of cytology, the study of cells. Anything with cyto in it is referring to cells. So cytology is the study of cells. Then along comes a guy about 5 years later named Anton van Leeuwenhoek. And Leeuwenhoek was looking at, for example, drops of pond water under the microscope. And he noticed all these little things swimming around. And his microscope seems even simpler, but it's actually able to magnify even greater than Hooke's microscope. And there are microscopes that you can get, and it's kind of a pocket microscope. So they copy Leeuwenhoek's microscope design to make pocket microscopes today. And again, they're surprisingly powerful, can really magnify a lot. But anyway, Leeuwenhoek, when he looked at pond water, drops of pond water under the microscope, he saw all these little things swimming around, and he called them animalcules. Kind of like molecules, or very, tiny things. So he called them animalcules because they were tiny animals. Today, of course, we call them microorganisms, or microbes for short. So Leeuwenhoek is basically the father of microbiology. Here we're looking at the development of cell theory. And cell theory is a relatively simple concept. But it took the work of these 3 scientists to come up with the idea. So the scientist that write is Matthias Schleiden. So in 1838, he was observing plant tissue under the microscope. And he collected samples from a lot of different plants. So this is just an example. And by the way, this is the same species that you'll be looking at under the microscope in a lab that we're going to do in this unit. So this is Elodea, which is an aquatic plant. But what's nice about it is the leaves are only about 1 cell thick. So the light goes right through the cells, the light goes right through the tissue, so you're able to see the individual cells and actually see inside the cells. So this is about at 100 times magnification. And this is similar to what Schleiden would be looking at under the microscope. But what he established by looking at all these different plant tissues under the microscope, he basically collected samples of plant tissue, every plant he could get his hands on, and looked at them under the microscope, and every single one was made up of cells. So he concluded that all plants are composed of cells. Schwann here, Theodor Schwann, did the same thing, but with animals. So he got a hold of all kinds of animal tissue and looked at it under the microscope. And he, in all the animal tissues he was looking at, he saw cells. Now, it's a little bit more difficult to see animal cells because they tend to be smaller. Plant cells, this is at 100 times magnification. What we're looking at here is pancreatic tissue. So it's tissue from the pancreas. And each little purple dot here is the nucleus of the cell. So we can't really see the outlines of the cells very well, the plasma membrane that surrounds the cell. In plant cells, it's easier to see the outline of the cell because they have a cell wall in addition to their plasma membrane. So it's a lot easier to see and delineate individual cells. But again, this is at 400 times. So that's even higher magnification, and the cells don't even look as big as the cells that we're seeing here, the plant cells that we're seeing at 100 times magnification. So my point here is that animal cells are a lot smaller than plant cells. But anyway, Schwann established that all animals are composed of cells. So now we know that all plants are composed of cells, all animals are composed of cells. So those are two parts of cell theory. All living things that's basically one part of cell theory. All living things are composed of cells. And don't worry, we'll list the three components of cell theory in a minute. But here we see Rudolf Virchow, and in 1855, he was observing cells under the microscope. And these are drawings that he made from the cells that he observed. And so maybe you can tell that what's going on here is cell division. So he established that all cells come from pre-existing cells. And that's another component of cell theory. And he termed it omnis, in Latin, omnis cellula e cellula. All cells from cells. So cell theory, to boil it down for you here, cell theory is that all living things are composed of cells. Cells are the basic unit of structure and function. And cells can only come from pre-existing cells. And that's what we now refer to as cell theory. But these are the three scientists that were involved in developing that basic tenet of biology. And as we go on to the next slide, keep in mind how small the nuclei of these animal cells are, even at 400 times magnification. Because we're going to be looking at a cell nucleus on the next slide, but it's going to appear much different than the nuclei that you see in this micrograph. So technology tends to evolve, and it's the same with microscopes. Microscopes have improved over time greatly, and all kinds of different kinds of microscopes have been invented to help us visualize all kinds of different things within cells. So there are two major classes of microscopes, the light microscope and the electron microscope. The first microscopes, of course, were light. Electron microscopes were developed much later. So all the discoveries that we were just going over on the previous slides, those were all done with light microscopes. The electron microscope, however, because it's using electrons instead of light, are able to produce 1,000 times greater resolution and magnification than light microscopes. 1,000 times. So pretty much the limit for a light microscope is about 1,000 times magnification. So an electron microscope being able to magnify 1,000 times greater than 1,000 times, that's about 1,000,000 times. And again, there are newer electron microscopes that are even able to get further than 1,000,000 times in magnification. So you can imagine that it's really helped out a lot in allowing us to see very, very small things. The drawback for the electron microscope, though, is that the specimen has to be dead, because in the process of producing or preparing the specimen to view it under the electron microscope, the specimen is killed. So that's a drawback. The micrograph on the left is a highly magnified two-dimensional, and that's important in understanding the difference between these two different kinds of electron microscopes. A transmission electron microscope, TEM, gives you a two-dimensional view of whatever you're looking at. And what we're looking at here on the left is a two-dimensional view of just the cell nucleus. So again, if you go to the previous slide and realize how small those cell nuclei are at 400 times, we're looking at just a nucleus here. Not a whole cell, just the nucleus of a cell. That's extremely magnified. And here you can see what you've heard of previously, like in the lab that we did, concerning where ribosomes come from, for example, the nucleolus. Ribosomes being involved in protein translation, if you'll recall. So if you're wondering what the nucleolus is kind of like the nucleus of the nucleus. And some nuclei can have more than one nucleolus, as you can see here. But that's basically where ribosomes are made. Ribosomal RNA is transcribed there. So the point here, though, is that a transmission electron microscope is going to magnify, it's called transmission because the electrons are going through the specimen. So to prepare the specimen, it has to be a really, really thin slice. And of course, that results in the death of the cell. So this is not a living cell. It's in grayscale. These can be colorized, like that first image we saw on the first slide, but otherwise it's a grayscale image, and it's got to be dead. You can't see anything moving around or anything functioning because the cell is dead. Here we see a scanning electron micrograph, and it's three-dimensional. So that's one of the biggest differences between transmission and scanning electron microscopy is transmission is two-dimensional, whereas scanning is three-dimensional, and it's because it's bouncing the electrons off the outside of the cell. So we're not seeing inside the cell, we're seeing the whole cell. So this is one whole cell, and this is one whole cell, and these are the cell bodies, actually, of neurons, nerve cells. But these are the two different ways that you can see cells using these two different electron microscopes. And that's the point, that we've developed these two different electron microscopes, and it visualizes cells in much different ways. So microscope evolution has evolved and developed over time, and I just wanted to give you two more examples of microscopes that have developed from both light microscopy and electron microscopy. So on the left, this is a light micrograph, but it's a special kind of light microscope that uses lasers to visualize specific cellular structures. So in what we're looking at here, the blue are nuclei. So the blue is the nucleus of each one of these cells, and we're looking at a number of different cells here. And then the red around that is a certain component of what we call the cytoskeleton, which is something that you'll be learning about in this unit. The cytoskeleton is made up of two different kinds of fibers. So we're seeing one of those fibers in red and the other of those fibers in green. But this is like the skeleton of the cell. It helps to support the cell. It helps to anchor things within the cell. It helps to move things around within the cell. So this microscopic technique allows us to be able to see that, something that we normally wouldn't be able to see. And you'll notice that these are labeled HeLa cells, HeLa cells. And that's short for Henrietta Lacks, who died in 1951, but these cells were taken from her. They're tumor cells. She had cancer, and these are tumor cells that were taken from her, and they're still alive today. And there are samples of them all over the world being used for research, which is actually a controversial thing because they were taken without her knowledge, and they're now being used for all kinds of different things, and her family is seeking compensation. I think the case might be over now, but that's all a big hullabaloo around these cells. And then on the right, we see what's known as a scanning tunneling electron microscope. This is a view from a scanning tunneling electron microscope. So it's scanning, which again gives you kind of a three-dimensional image. But the tunneling here is because it's using a probe, a very, very fine probe. And I guess these probes can get down to the point where the tip of the probe is like one atom sharp. If you think of sharpness, getting smaller and smaller and smaller, the probe here is only one atom. And it traces the surface of samples and magnifies them much greater than what you can get with a regular scanning electron microscope. And so what we're seeing here is DNA, molecules of DNA. You can't see the individual nucleotides making them up, but it's incredible that we've got microscopes that are able to get the level of magnification necessary to see molecules. And I guess some of these microscopes have even been developed to be able to see individual atoms. Pretty incredible. So just how small are cells, or what is the size range for cells? You'll notice that this is similar to the hierarchy of biological organization. We start here with atoms and move up to molecules and larger molecules like proteins that come together to form cellular structures like ribosomes and organelles. And then we get to the simplest cells, the smallest bacteria, prokaryotic cells, and then eukaryotic cells, and onward. And you'll notice that the units that are being used to measure these things are changing. And so let's take a look at that first. NM stands for nanometer, and a nanometer is, if you're familiar with scientific notation, 1 × 10 to the -9th meter. So when you have a minus superscript here, or a negative exponent, that means that's the number, that indicates the number of places to the right of the decimal. So in other words, this is 0.00000000001 meter. That's an extremely small number. And it's 1/billionth of a meter. That's what 1 × 10 to the -9th is, 1/billionth of a meter. So that's the size of a nanometer. And that's what's used to measure things that are all the way down to the atomic and small molecule level or size. Then we have the micrometer. So as we go up here, we have nanometers until we get to here, and we have 1 micrometer. And a micrometer is 1 × 10 to the -6 meter. In other words, it's a millionth of a meter. It's 0.000001 meter. And that brings us to the millimeter, which is 1 × 10 to the -3rd meter. It's 1,000th of a meter, right? 0.001 meter. And then we get to the centimeter, which is 1 × 10 to the -2 meter, and then, which is 100th of a meter. And then the decimeter is 1 × 10 to the -1 meter. We don't use that much, but it exists, and it's 1/10th of a meter. And then, of course, we have the meter, which is the base unit. That's what all these other units are based on. So we need these very fine units of measure to be able to talk about and measure and communicate about the size of these very small things, including cells. So the realm of the prokaryotes, so to speak, ranges all the way down to the smallest bacteria, which are about 1/10th of a micron, all the way up to about 100 microns, or micrometers. And by the way, micrometer or micron is short for micrometer, and that's what you'll be using in the lab to measure the size of cells, or to estimate the size of the cells that you're looking at, is the micrometer or micron. But bacteria, prokaryotes, bacteria, those two words are synonymous. They mean the same thing. When you say prokaryote, think bacteria. When you think bacteria, think prokaryote. These are cells without a nucleus. They're the simplest cells. They were the first living things on the planet, and they're the smallest. And then we have the eukaryotes, which range in size from about 10 microns to 100 microns. And generally, the animal cells, eukaryotes include animal cells and plant cells, and the animal cells are down in the range from 10 microns to 50 microns, where the plant cells tend to be larger, ranging from about 50 microns to 100 microns. And this also includes, other eukaryotes include fungi and protists. So fungi and protist, fungi, animals, and plant cells are all eukaryotic cells, and they range in this size range. So a reminder, this is our introduction to cells, and I want to remind you that all living cells have certain components. They all have DNA, they all have ribosomes, they all have cytoplasm and a plasma membrane. So those are things that all cells share. But here we're going to, well, what I asked you to do was draw an example of a prokaryotic cell on the left and a eukaryotic cell on the right and label it with those components or more. So you should have hand drawn this. I went ahead and found drawings instead of using my own drawings, but these are hand drawn. And then we want to point out some characteristic differences between prokaryotic cells and eukaryotic cells. But notice that what's labeled here are basically what all cells share. So there's DNA. The nucleoid here is referring to the DNA, where the region where DNA can be found in a prokaryotic cell. This plasmid is also a little ring of DNA. So all cells have DNA. You'll notice that the cell membrane, and we're going to correctly call it the plasma membrane, but the cell membrane in this diagram and the plasma membrane are the same thing, and all cells have a plasma membrane. Not all cells have a cell wall, however, so don't misconstrue that as meaning all cells have a cell wall. Not all cells do. Prokaryotic cells do. And plant cells do, and some protists do, but not all cells do. All cells have ribosomes because all cells need to be able to make protein, right? If you'll recall, ribosomes are the site of protein synthesis, the site of protein translation. You just learned all about that. So all cells have ribosomes because all cells need to be able to translate proteins. PLI, that's specialized to prokaryotic cells. And we'll come back to that, though. You do need to know what that is, and flagella also in this unit. You do need to know what those are. But anyway, prokaryotic cells don't have a nucleus and membrane-bound organelles. They don't have a membrane-bound nucleus, and they don't have membrane-bound organelles. They were the first cells that evolved about 4 billion years ago, simplest and first. As we learned from the previous slide, they range from about 1 micron to 10 microns, but they can be all the way down to 1/10 of a micron in size. No membrane-bound nucleus, no membrane-bound organelles. And they're represented by the domains, because there are 3 domains. All life has been classified into 3 domains. Archaea and bacteria are the prokaryotic domains. And they are also kingdoms. The domains are also kingdoms. So Archaea is the kingdom Archaebacteria. The domain Archaea is the same as the kingdom Archaebacteria, and vice versa. And the domain Bacteria is the same as the kingdom Eubacteria. So you need to try to keep those names straight. In other words, if I ask you for the domain of bacteria, you need to say bacteria, and if I ask you for the kingdom of a bacteria, you need to use the term eubacteria. Here we see a eukaryotic cell, and as you can see, it's much more complex. This is a plant cell. Plant cells actually have more organelles than animal cells do. So actually, we're going to start with plant cells in our study of eukaryotic cells when we get there, but they evolved about 2 billion years ago. So notice that prokaryotes were on the planet for 2 billion years before eukaryotes evolved, approximately. And if you'll recall, because we studied this in previous units, endosymbiosis is the theory that explains how eukaryotic cells evolved from prokaryotic cells, if you'll recall. And it's basically the idea that mitochondria and chloroplasts that we find in eukaryotic cells are started out as endosymbionts. They started out as symbiotic organisms living inside other prokaryotic cells. So they are prokaryotic cells that were living inside of other prokaryotic cells, and that partnership became eukaryotic cells, is the basic idea, if you'll recall. They're, as we learned on the previous slide, 10 to 100 microns in size. Animal cells, again, tend to be smaller than plant cells. They have a membrane-bound nucleus. That's one of the major contrasts from prokaryotes. No membrane-bound nucleus or organelles. Eukaryotic cells have a membrane-bound nucleus and organelles. The domain is only one domain. All eukaryotes are in the domain eukarya. So again, there are three domains, archaea, bacteria, and eukarya. Two, archaea and bacteria, are prokaryotic, whereas the domain eukarya are all the eukaryotes. And there are four kingdoms within domain eukarya. There's the kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. And three of those are primarily multicellular. Fungi, Plantae, and Animalia Plantae and Animalia are all multicellular. Fungi are mostly multicellular, and there are even some multicellular members of the kingdom Protista. But most of Protista are unicellular organisms. But anyway, that's the difference between these two cell types, prokaryotes versus eukaryotes. And the final point I want to make here is that prokaryotes were first. By about 2 billion years, prokaryotes were first, and they set up the system that we all live within. An example of that is that some of these prokaryotes were photosynthetic, cyanobacteria, and in the process of photosynthesis, they released oxygen, and they're responsible for oxygenating the earth, oxygenating the planet. The oxygen you're breathing in the air right now was first put into our atmosphere and is still being maintained in our atmosphere by cyanobacteria.